All right, so how do we get from just these like super simple single-celled organisms all the way to, like everything around us,
Right.
all the plants and animals, and us too? It's one of those really big questions in biology, right? And it all started with just looking around, seeing the incredibly small.
Yeah, it's fascinating when you think about it. Leuwenhoek back in the 17th century, and with his little microscopes, he's looking at the stuff for the first time. He sees bacteria, describes them as incredibly small alongside all these other little animals.
Right, and it's like that's the first time anyone had ever really seen this hidden world.
Exactly. It really was like opening this whole new chapter in biology. And what's amazing is that even with his pretty basic instruments, like compared to what we have today, he still kind of had this gut feeling that the bacteria were different from those bigger protists he was seeing.
Oh, so he was already making that distinction.
Yeah, like he even called the protists gigantic monsters compared to the bacteria. So even way back then, people were starting to see that there were these different scales and types of microscopic life.
And he was finding bacteria everywhere, right?
Yeah.
Like it wasn't just one weird sample he was looking at. He was in rainwater and seawater and, even the plaque on his teeth.
Oh, yeah, he found them all over the place. And, he also noticed that some of the larger cells had these little globules inside them, which we now know were most likely the nucleus.
Wow. So he was really observing some pretty fundamental stuff.
Yeah, he was laying the groundwork. But, it's interesting because for a while after that initial discovery, things kind of stalled out. Like, we had these snapshots of the microscopic world, but the bigger picture was still blurry.
So it's like we knew they were there, but we didn't really know what to do with that information.
Right. And, about 50 years after Linnaeus came along, and he kind of just lumped all these microscopic organisms together into this vague category called chaos.
Chaos. I mean, that kind of sounds about right.
Yeah.
It must have been overwhelming to see all that diversity for the first time.
I know, right? So it wasn't until the 19th century with Ernst Haeckel that we started to see a real effort to separate bacteria from other microbes again. So even though we could see these organisms, it took a while to really grasp how they fit into the grand scheme of life.
It's almost like we had to develop the right conceptual tools to make sense of it all.
Exactly. Seeing something and understanding it are two very different things, and that's why those later discoveries about genetics and cell biology would turn out to be so revolutionary.
And that's exactly why we're here today, to really delve into this question of how complex life arose from those simpler forms. How did we go from bacteria to everything else?
Exactly. And in this deep dive, we're going to explore those major revolutions in our understanding, focusing on the relationship between bacteria and the evolution of those complex cells that make up you and me.
And we're not going to get too bogged down on the jargon, right? We want to make sure everyone can follow along and really grasp the big picture.
Right. It's all about those key insights and the surprising facts that make you go, aha, I get it.
Exactly. So our mission is to unpack that monumental evolutionary leap from those simple bacteria to all the complexity we see in eukaryotic life.
Right, because eukaryotic life is the kind of life with cells that have a nucleus like ours. Plants, animals, fungi, all that good stuff.
All right, so let's go back to the beginning with that first revolution, Leeuwenhoek's discovery, and those early classification attempts.
Right, so we've touched on his observations of bacteria being incredibly small and how he sort of instinctively knew they were different from the larger protists.
And it's important to remember just how widespread his findings were. These things were everywhere.
Oh yeah, he was finding them in all sorts of places, and even without the tools of modern cell biology, his observations were hinting at these fundamental differences between these microscopic things.
So like what kind of hints?
Well, he described these globules within the larger cells, which, as we said, were probably nuclei, and he even noted their size relative to the free-living bacteria. So it's like he was already picking up on these different levels of organization within cells.
Oh, well, that's pretty impressive for the time.
It really is. And then you have Linnaeus and Haeckel trying to bring some order to this whole new microscopic world.
And Linnaeus just went with chaos, right?
Yeah.
Like it was all just too much to handle.
Yeah, it kind of seems that way, right? But to be fair, it must have been completely mind-blowing to see this incredible diversity under the microscope for the first time. So this broad categorization was an attempt to start making sense of it all.
And then Haeckel comes along and says, hold on, maybe we can separate these bacteria out again.
Exactly. And that was a huge step forward because it acknowledged that there was a deeper evolutionary division, even though we didn't fully understand it yet.
So that brings us to revolution number 2, the ancestry of genes. Now, Francis Crick, even back in 1958, had this incredible prediction about protein taxonomy.
What's really cool is that Crick was way ahead of his time. He understood that the sequences of amino acids that make up proteins weren't just there for function, they were also like a historical record of evolution.
So he was saying that by comparing the sequences of these proteins, we could actually trace the relationships between different organisms.
Exactly. He called it the most delicate expression possible of the phenotype, meaning that these tiny variations in protein sequences could reveal those evolutionary connections much more precisely than just looking at physical characteristics.
And then, of course, we figured out DNA and how it codes for those protein sequences.
Right, and that gave us an even more powerful and sensitive tool to explore those evolutionary relationships. It was like going from a blurry photograph to a high-definition image.
So it was like finally seeing the tree of life in sharp focus.
Exactly. And that brings us to a really exciting point, because now we're talking about a third cellular revolution, one that's building on those first two, and even challenging some aspects of Carl Woese's famous tree of life.
Okay, so what was this tree of life? That was based on comparing the sequences of a specific gene, right? To show how the three domains of life, bacteria, archaea, and eukaryotes, are related.
Right, and his work was groundbreaking, but what's happening now is that researchers are starting to look at the bigger picture. They're asking how the physical cells themselves and their genetic information actually relate in terms of evolution.
So it's like going beyond just looking at one gene and trying to understand the whole cell as a product of evolution.
Exactly. And this is where things get really interesting, because we have these two kind of contrasting ideas. One is divergence, which is all about things splitting apart over time.
Right, like branches on a tree.
Exactly. And that's what Woolwe's Tree of Life emphasizes. But then we also have convergence, which is about things coming together.
And that's where Lynn Margulis and her endosymbiotic theory come in, right?
Exactly. Margulis was all about how genes could come together through symbiosis, the physical merging of different organisms living in close association.
So it's like two sides of the same coin, divergence and convergence,
Yeah.
both shaping the evolution of life.
Right. And what's becoming increasingly clear is that the real answer lies somewhere in between these two ideas.
So it's more complex than just one or the other.
Oh yeah, it's much more nuanced and a lot more exciting. We know now for sure that key components of complex cells like mitochondria and chloroplasts, they definitely came from bacteria through endosymbiosis.
Right. Mitochondria, those are like the powerhouses of our cells.
Exactly. They're responsible for producing most of the ATP that our cells need to function. And chloroplasts, they're the organelles that allow plants and algae to perform photosynthesis.
And both of those came from bacteria.
That's right. It's pretty wild when you think about it. But here's the thing. Chloroplasts seem to have been acquired later in evolutionary history, specifically in the lineage that led to plants and algae.
So plants got their chloroplasts later than other eukaryotes got their mitochondria.
Exactly. Mitochondria are much more ancient. They were present in the common ancestor of all eukaryotes.
And, I remember learning in textbooks that it was like this sophisticated amoeba-like cell that just kind of engulfed a bacterium and bam, mitochondria. But you're saying that's not quite right.
Well, that's the traditional story, but more recent genetic evidence has really changed our understanding. The host cell that first acquired the mitochondrion wasn't a complex eukaryotic cell at all. It was an archaeon.
An archaeon. But archaea are like bacteria, right? They're prokaryotes. They don't have a nucleus.
That's right. So this was a huge discovery because it meant that complexity didn't necessarily arise from other complex cells. It was this unexpected partnership between a simple archaeon and a bacterium that paved the way for eukaryotic life.
So it's like this really ancient merger that set the stage for everything that came after.
Exactly. And how that actually happened, the mechanics of it are still being actively researched. It's a really fascinating puzzle.
So wait a minute. Archaea don't even have the machinery to engulf other cells, right? Like they don't do phagocytosis.
You're right. That's one of the big mysteries. How did this simple archaeon without a nucleus, without phagocytosis, managed to acquire something as complex as a mitochondrion? It seems like a huge evolutionary leap.
It's mind-blowing. And that brings us to the really exciting stuff that you've been digging into, this idea that it wasn't just about genes or the environment. It was also about these fundamental energetic constraints that shapes the evolution of life.
That's right. This is one of the most intriguing arguments being made right now. It suggests that energy might be even more important than we previously thought in determining why life on Earth evolved the way it did.
So it's like looking beyond just the information in DNA and consider the energy that's needed to make it all work.
Exactly. And it ties into Erwin Schrödinger's famous book, What is Life? Remember he talked about how life seems to defy the second law of thermodynamics?
Right, like how life manages to stay organized and resist that natural tendency towards disorder and entropy.
And he saw genes as holding this crucial code script for life.
But he thought it was proteins that carried the genetic information, not DNA.
Right. So that's interesting to consider because the later discovery of DNA really solidified this information-centric view of biology.
Yeah.
We became obsessed with deciphering the code script.
And maybe we overlook the energy side of things a little bit.
It's possible because the code is just information. It's not enough to explain the incredible complexity of life. You also need the energy to read that code, to interpret it, and to make it all work.
And genomes are really complex.
Yeah.
Right, like we have billions of DNA letters, but only a small fraction actually code for proteins.
Exactly. And there's this really weird thing where there's no clear correlation between the size of an organism's genome and its complexity. Like onions have way more DNA than humans.
So you can't just say that more DNA equals more complexity.
Right. It suggests that other factors must be at play, things like the efficiency of energy utilization and the overall architecture of cells.
So energy efficiency is a major factor in complexity.
It seems that way. And it all points to this idea that we can't just look at information alone. We need to consider the energy budget of life.
Right. And you mentioned isotopic fractionation as one of the earliest pieces of evidence we have for life in ancient rocks.
Yeah, isotopic fractionation is really cool. It's basically about how biological processes tend to prefer lighter isotopes of certain elements.
So like different versions of the same element with slightly different weights.
Exactly. And living things often prefer the lighter ones, which leaves a distinct chemical signature in rocks. So we can look at these really old rocks and see evidence of this isotopic fractionation.
And that suggests that life was present.
It's a good indicator, but there's a catch. Similar patterns can also happen through purely geological processes, especially in environments like hydrothermal vents.
So it's not a foolproof sign of life.
Right, it's more like a clue that hints at the possibility of early life. It's hard to draw a definite line between non-living and living in those ancient environments.
It's probably more of a continuum, right? Like these geochemical processes might have been the precursors to life.
Exactly. The early Earth was a wild place full of geological activity, and the processes that led to life were probably deeply intertwined with those forces.
So it's like a blurry boundary between the non-living and the living.
Exactly. And speaking of the early Earth, we know that bacteria played a huge role in shaping its environment.
Oh yeah, like those banded iron formations, right? Those are absolutely stunning.
They really are, and they were created by bacteria. These formations are basically layers of iron oxides, and they tell the story of how early bacteria literally rusted the oceans.
So how did bacteria rust the oceans?
Well, they do it through their metabolism. They were stripping electrons from dissolved iron and leaving behind these layers of rust.
And that's what created those beautiful bands of red and black.
That's right. And those iron deposits are actually connected to early forms of photosynthesis.
Oh, wow. So photosynthesis has been around for a long time.
Oh, yeah. But those early versions were different from what we see in plants today. They were more primitive, and they used different electron donors like dissolved iron or hydrogen sulfide gas.
So instead of releasing oxygen as a byproduct, they were releasing rust or sulfur.
Exactly. It wasn't until later that oxygenic photosynthesis evolved.
And that was a major turning point, right?
Huge. Oxygenic photosynthesis, which probably evolved between 2.9 and 2.4 billion years ago, allowed life to use water as an electron donor and release oxygen as a byproduct.
And that led to the Great Oxidation Event, which really changed the planet.
Oh yeah, that was a game changer. The atmosphere became oxygenated, iron on land started to rust, and it even triggered those snowball Earth glaciations by oxidizing methane, which is a potent greenhouse gas.
So oxygen really reshaped the planet.
It did. And for a long time, the traditional narrative was that these rising oxygen levels were the main driver of biological innovation.
Like oxygen was the key to unlocking greater complexity.
Exactly. But one of the sources you shared actually challenges that view. It suggests that maybe the structure of cells themselves was even more important.
So it's not just about the environment, but about the internal constraints of cells.
Right. And it's not to say that oxygen wasn't important, but maybe it wasn't the only factor driving complexity.
So what kind of evidence supports that idea?
Well, if oxygen was the main driver, we'd expect to see these rapid bursts of diversification whenever oxygen levels increased. But what we actually see in the fossil record is a bit more complicated. There are long periods of relative stability with occasional big jumps in complexity.
So it's more like these bursts of innovation happening when some internal barriers overcome.
That's the idea. And one of the biggest barriers might have been the evolution of complex multicellularity.
Right, because you need a lot of coordination and specialization for cells to work together as a single organism.
Exactly. And that seems to have been a major hurdle that was only overcome once in the lineage leading to animals.
So it's like these structural limitations within cells were just as important as environmental factors in shaping evolution.
That's the argument. And this is where Lynn Margulis and her serial endosymbiosis theory come in.
Right. She was all about these dramatic mergers between cells creating new, more complex entities.
Exactly. Margulis was a real visionary. She argued that eukaryotic cells didn't evolve just through gradual Darwinian selection, but through these big jumps where one bacterium essentially moved into another cell and they started working together.
And we know that's how mitochondria and chloroplasts originated, right?
That's right. Those are the textbook examples of endosymbiosis. But Margulis went even further. She suggested that other eukaryotic organelles, like the nucleus, might have also originated through symbiosis.
Wow, so the whole eukaryotic cell might be a collection of former bacteria.
That was her idea. And it's called serial endosymbiosis because it would have happened in a series of steps over millions of years.
And here's something that I find absolutely mind-blowing. All eukaryotes, from a tiny yeast cell to a giant blue whale, we all share a single common ancestor.
That's right. All eukaryotic life can be traced back to one single origin event billions of years ago.
That's incredible, and it makes you wonder why it only happened once.
Exactly. It suggests that there were some very specific and maybe even rare conditions that had to be met for this to happen.
So it wasn't just about having the right ingredients, it was about overcoming some really fundamental barriers.
Right. And that's where the book circles back to its main argument that information alone isn't enough. We also need to consider the role of energy.
So it's not just about having the DNA blueprints, it's about having the energy to make it all work.
Precisely. And that's where Schrödinger's footnote becomes really interesting. He mentioned that free energy would be crucial for understanding life from a physics perspective.
So he was already hinting at this connection between life and energy.
He was. And by adding energy to the equation, we shift our focus from just what is life to what is living.
It's about those active processes like growth and metabolism and reproduction.
Exactly. And what's really amazing is that the core mechanisms for harvesting energy are incredibly similar across all forms of life.
So whether you're a bacterium or a blue whale, you're using the same basic principles to get energy.
That's right. It suggests that these energetic processes impose some very fundamental constraints on all cells. And we're still learning about how those constraints shape the evolution of life.
And it's fascinating to think about this continuum between non-living and living, like viruses or bacterial spores, or even those tardigrades that can survive in crazy conditions.
Those are great examples. They really blur the lines of what it means to be alive.
Even the NASA definition of life as a self-sustaining chemical system capable of Darwinian evolution gets tricky when you try to apply it to things like viruses.
Definitely. And it highlights this idea that living isn't a simple on-off switch. It's more like a spectrum.
It's about the interaction between a cell's internal structure and its environment.
Exactly. We know a lot about how genes encode the building blocks of cells, but we're still learning about how those physical constraints and the need for energy shape the overall structure of life.
So it's like we're starting to appreciate the importance of physics and energy alongside genetics.
That's right. And it brings us to the real heart of the matter, the central role of energy and those incredible proton gradients in powering life.
Okay, so lipid membranes. Those are the boundaries of all our cells. They're made of these fat-like molecules, and they form spontaneously, right?
Yeah, it's all about the hydrophobic effect. Those lipid molecules don't like water, so they arrange themselves in a way that minimizes their contact with it. And that creates a bilayer, a double layer of lipids.
And that's a low-energy structure, right? It's actually more stable that way.
Exactly. But life needs energy to do work, to grow, to move, to replicate. And that's where Gibbs free energy comes in.
So Gibbs free energy, that's the energy that's available to do work.
Right. And ATP is like the universal energy currency of cells. It's what powers all those biological processes.
And we get ATP through respiration, right?
That's one way. And respiration is all about transferring electrons ultimately to oxygen.
Like burning fuel, but in a controlled way.
Exactly. It's like a carefully orchestrated series of steps where electrons are passed down a chain of molecules, releasing energy along the way.
And that energy is used to pump protons across a membrane.
That's right. It creates a proton gradient, a difference in concentration of those positively charged hydrogen ions. And that gradient stores potential energy.
And that's what powers the ATP synthase.
Exactly. The ATP synthase is like a tiny rotary motor. It uses the flow of protons back across the membrane to generate ATP.
It's like a hydroelectric dam, but on a molecular scale.
That's a great analogy. And it's mind-blowing because this ATP synthase is found in all forms of life, bacteria, archaea, and eukaryotes. It's a universal enzyme essential for life.
So it's like a fundamental piece of the puzzle.
It really is. And it all goes back to Peter Mitchell and his chemiosmotic hypothesis.
Which wasn't immediately accepted, right? It was a pretty radical idea at the time.
Yeah, people thought ATP synthesis had to involve a direct chemical transfer of a phosphate group. But Mitchell came along and said, no, it's this proton gradient that drives it all.
And he was right. It's amazing how sometimes those unconventional ideas turn out to be the most groundbreaking.
It's true. And this whole reliance on electron transfer and proton gradients really highlights the importance of kinetic barriers.
It's like how oxygen and methane can exist together without immediately exploding.
Exactly. They're thermodynamically unstable, but they're kinetically stable, meaning there's a barrier preventing them from reacting immediately.
And life takes advantage of those barriers.
Right. Absolutely. It uses enzymes as catalysts to control and speed up specific reactions, extracting energy in the process.
So life is like this master manipulator of energy, always finding ways to get what it needs.
That's a great way to put it. And it brings us to the incredible metabolic versatility of bacteria and archaea.
Right, they can use all sorts of different electron donors and acceptors depending on what's available in their environment.
They're like metabolic ninjas, able to adapt to almost any energy source.
And they can swap genes for those energy-related proteins through lateral gene transfer.
Exactly. They can share those genes with each other, which allows them to adapt really quickly to new environments.
So it's like they have this redox protein construction kit that they can mix and match to suit their needs.
That's a great analogy, and it all leads to this exciting possibility that by understanding these fundamental energetic principles, we might be able to develop a more predictive biology.
So we might be able to predict what kind of life could exist on other planets based on their chemistry.
That's the idea. It's a long-term goal, but it's really exciting to think about.
So let's recap our deep dive today. We started with the discovery of the microscopic world and those early classification attempts.
Right. Then we explored the phylogenetic revolution and how understanding the ancestry of genes gave us a whole new perspective on the tree of life.
And finally, we delved into this emerging view that energy and endosymbiosis were crucial drivers in the evolution of complex life.
And we saw how bacteria played a key role, not just as separate entities, but as the very source of some of our most important cellular components.
And that the host cell in that first endosymbiotic event was likely a simple archaeon, which is a pretty wild twist.
It is, and we're still trying to unravel the mysteries of that ancient merger and the energetic constraints that made it all possible.
And one of the most fascinating things for me is this idea that at its core, all life might be about managing the flow of electrons and harnessing the power of those proton gradients.
It's a beautiful concept, right? This fundamental process that connects all forms of life from the simplest bacteria to us.
So here's a final thought for you to ponder. If the origin of complex life was so heavily influenced by these specific energetic constraints and that rare endosymbiotic event, what does that say about the likelihood of complex life evolving elsewhere in the universe?
Right. Does the universality of these energy principles and the ATP synthase suggest that similar solutions might arise on other planets? Or was the path to complex life a one-time fluke, a series of lucky events that might be incredibly rare in the cosmos?
It really makes you think about the interplay of necessity and chance in the grand scheme of things. And it leaves us with more questions than answers, which is exactly what makes science so exciting.
I completely agree. It's the mystery that keeps us exploring and pushing the boundaries of knowledge.
Absolutely. Thanks for joining me on this deep dive into the origins of complex life. Until next time, keep those brains buzzing.
It's been a pleasure.
And to all our listeners out there, thanks for tuning in. We'll see you on the next deep dive.